Hello, my name is Anwing, and today I'll be discussing Toxoplasma gondii, a microscopic parasite, and it's also one of the most widespread organisms on Earth. So, Toxoplasma gondii is a unicellular eukaryotic organism belonging to domain Eukarya, phylum Apicomplexa, class Conodicida, order Eukaryotes, Diorita, family Sarcocystidae, and genus Toxoplasma. And although there's only one, T. gondii is only like the only one in its genus. There are also other kinds and strains of T. gondii, though there aren't as researched. And unlike bacteria, T. gondii possesses a nucleus and other membrane-bound organelles typical of eukaryotic cells. And these parasites have a distinctive crescent shape and around 6 micrometers in length approximately, but it varies, and they're an obligate intercellular parasite. So one of the most important features about it is its apical complex, a specialized structure that allows parasite to invade host cells effectively. So we're going to talk about the different shapes it takes, because T. gondii exists in three major forms throughout its life cycle. Eukaryotes, the first one, is responsible for acute infections in their rapidly reproducing forms, while the other form, Bradyzoites, you can see right here, they're a bit smaller and they grow a lot more slowly and remain enclosed within tissue sites during chronic infections. And you can sort of see what the tissue sites looks like here. Again, crescent shapes. And finally, sporozoites are formed inside environmentally resistant oocysts produced by cats. And we'll talk about that more later, but this is it here. And they are very resistant, and this is the one that survives the outside of a host, essentially. And once inside the cell, the parasite creates a specialized incarnate called a parasitophorous vacuole, and this protects the parasite from the host while providing an environment where it can grow and reproduce. And you can start to see it here where the parasite will invade the host cell and then form this vacuole here where it will start to replicate and then burst once it is done. And these hosts include humans, birds, rodents, and livestock, other different kind of livestock, where the parasite reduces asexually and reproduces asexually. And during the tachyzoid stage, reproduction occurs very rapidly, allowing the infection to spread throughout the body. And as the immune system responds, the parasite often converts to bradyzoites and forms tissue sites. And these sites can remain dormant inside the body for years, especially in muscle tissues and the eyes and the brain. However, the one organism where T. gondii reproduces sexually are cats, as cats can support the parasite's sexual reproductive stage, and in this stage, oocysts are produced and shed in cat feces. And this is where oftentimes humans come in contact with the with the parasite, and recent research have also shown that the infection can alter the composition of people's gut microbiome, and scientists have observed certain increases in bacterial species and decreases in other, including lactobacillus, resulting in disruption in normal metabolic processes. And one interesting aspect of its biology, T. gondii's biology, is the relationship between cats and rodents, as research shows that infected rodent may become less fearful of these cat odors, and it's more likely they could be eaten for cats, and this parasite will complete its life cycle by returning to the one that can actually help it sexually reproduce. And for most healthy individual. Infection from this parasite causes like a mild flu or no symptoms at all, according to the CDC. However, the parasite is more dangerous towards immunocompromised people, such as those with HIV, and in this case, the dormant cyst may actually reactivate and cause more severe complications like damage to the brain and nervous system. And another major concern is congenital toxoplasmosis, where a pregnant individual becomes infected and the parasite can be transmitted to the baby. And this is why pregnant women aren't recommend against skipping cat litter. And so because of these risks, toxoplasmosis is an important health concern. But overall, it's a very successful parasite with a complex life cycle. I think that's the most interesting part about it. And it has like a good ability to invade host cells and evade immune defenses. Yeah. And thank you for listening. This is my works cited.
The microbe that I have chosen for this assignment is the pathogenic fungi, a brain-manipulating fungus known to alter the behavior of an ant host. The name is Eukaryotes unilaterus, and it is specific to different ant tribes. The organism is found throughout the tropical and subtropical regions of both the eastern and western hemispheres. These fungi, which include both sexual and asexual stages, utilize a specialized DNA genome to control host behavior, often modulated by internal RNA microviruses. The fungus compels the ant to descend to a humid microclimate favorable for fungal growth, typically about 10 inches off the ground. Upon reaching the underside of the leaf, the north side of the plant, the ant sinks its jaw into the leaf, securing itself in a permanent death grip. While the ant is anchored, the fungus consumes its victim's internal organs. Within 7 days of the death's ant, the fungus begins its final reproductive stage. A fungal stroma, or fruiting body, grows and erupts through the back end of the ant, turning the shriveled corpse into a launch pad. Finally, the pad develops into a stroma, allowing the fungus to jettison its spores into the air to infect a new generation of foraging ants below. O. unilaterus does not act alone. It relies on complex bacterial symbiosis, notably with bacillus bacteria, that operate alongside the fungus. These bacteria co-occur and synergize with Eukaryotes, actively producing toxins, degrading host tissues, and protecting the fungal network from competitors as it consumes the host. For the art part of my project, I decided to make clay figurines. And he's biting in. For the art part of my project, I decided to make clay figurines, kind of showing what the process was for the ants and the fungus. So here you can see I made the ant, and he's biting into the leaf, and on the end is the stroma, or the cushion there, coming out from the back side. And then I decided to do a close-up of the cushion. As you can see, it's got these kind of sharper projections here. That's what it looks like close up. You can see it's got the it's like getting ready to release its spores there. Then I decided I'll do a close-up of that. If you cut the cushion here in half and enlarge it, this is kind of what it looks like. You can see there's lots of cavities inside. The little spores are along the edge here. These are what will eventually release. So that's the section through this, which is that guy there on the ant. And then finally, when the spores are released about a month later, this is what they look like. And you'll see there's a little protrusion on the edge. On the end there, the white spot, and it's ready to infect another ant. So that's the stages. We've got the ant, bites the leaf. That's a close-up of the stroma, the base there, the cushion. This is the cushion cut in half, little spores, the cavities inside, that's how it's about to burst and release its little spore here.
Eustylagomeatus is a fungus, also known as corn smut. Anatomically, you can see that it creates these big galls on corn. On a microscopic level, it creates teleospores, which are released. They're winter resistant, and each of those galls can produce 25 billion spores. Those spores will then become haploid sporidia. They're cigar-shaped, and those then become dikaryotic filaments, which go into the plant. Those anatomically are actually used as a model for human neurons. So Eustylagomeatus is also known as huitlacoche. It's an indigenous food preparation. It comes from two Nahuatl words for black and excrement. It co-evolved alongside corn and also its ancestor Teosinte. The phylogeny of Eustylagomeatus is interesting in that it is not actually as related to the genera Eustylago as it is other genera such as Sporosorium and Anthracositis. It is sometimes referred to as Mycosarcoma meatus. This is a simplified diagram of its life cycle. Those teleospores that we talked about earlier are step 1. They come from existing Eustylagomeatus, and they then germinate in order to become those cigar-shaped haploid cells. And those haploid cells are saprobic, meaning they eat dissolved organic matter to become dikaryotic filaments, which then infiltrate the extracellular space of that corn plant. That's where these tumors are formed on the corn. So the metabolism of Eustylagomeatus is interesting in that galactose is toxic to it, so it doesn't use the oxidoreductive pathway that other organisms use. It uses another shorter pathway that is mediated by different enzymes. It's those GAL enzymes below, which can then lead to two different products, either a new glucose or a new galactose. And if it's the glucose, it will either be used for glycolysis or glycogen synthesis based on the needs at the time. If it is the galactose, it will be used as a precursor for glycolization. So endohyphal bacteria, or EHB, are a specific type of bacteria that are symbiotes with fungi. They actually live inside of fungi, and they have benefits such as increasing the number of spores that it produces. Specifically, Klebsiella michiganensis has been found inside of Eustylagomeatus, and there's evidence that they co-evolved because there are a lot of bacteria that don't contain this enzyme chitinase, and those are not found in fungi because they cannot penetrate the fungal cell wall. So here is a simplified diagram of that cell wall. The orange part is the chitin, and in the second part of the image, the bacteria is releasing chitinase, and that actually breaks through the chitin and allows the bacteria to enter the cell. And it's interesting that there's barely any trace that it will have entered, and essentially no damage to the fungus. So Eustylagomeatus produces clusters of reproductive bodies, also called sori, which eventually rupture into those black masses. And it also secretes effectors that do a lot of the pathogenic work. They can disrupt signals, metabolism, and immunity of the host. And like we mentioned earlier, those filamentous growths are what actually infiltrate the plant host. So overall, Eukaryotes does not cause a lot of crop loss compared to other fungi. It does actually become another food in and of itself, that Eukaryotes. And besides infecting corn, it can infect grasses. But I think its most interesting usage is probably its usage in tacos and quesadillas.
For my presentation, I chose malaria. My artwork will depict the life cycle of Plasmodium, the protozoan parasite responsible for malaria, which is one of the most persistent and deadly infectious diseases in human history. The word malaria comes from the Italian term malaria, meaning bad air. Early populations associated the illness with swampy environments. We now understand that malaria is actually a vector-borne parasitic disease transmitted through the bite of infected female Anopheles mosquitoes. Malaria remains a global health issue. In 2024 alone, there was an estimated 282 million cases and over 610,000 deaths worldwide. About 95% of these deaths occur in sub-Saharan Africa, and children under 5 account for the majority of fatalities. Despite decades of prevention efforts, malaria cases have recently increased due to climate change expanding mosquito habitats, growing resistance to insecticides, and parasite resistance to anti-malarial drugs. Plasmodium has a really extraordinary life cycle as it occurs both in mosquitoes and humans. I'll specifically be talking about Plasmodium falciparum. So the life cycle begins with the female Anopheles mosquito feeding on an infected human. Only females transmit malaria because they require blood meals for egg development, so males do not. During feeding, the mosquito ingests specialized parasite called gametocytes, and this is the sexual stage of Plasmodium. Inside the mosquito's gut, these cells fuse to form a zygote, which develops into a mobile form called the eukaryote. The eukaryote penetrates the mosquito's gut wall and forms an oocyst, where hundreds of infectious sporozoites develop. After about 1 to 2 weeks, these sporozoites migrate to the mosquito's salivary glands, preparing the mosquito to infect its next human host. When this infected mosquito bites another person, it injects sporozoites into the human host. It enters the bloodstream through the mosquito's saliva. So now the vector has changed from mosquito to human. The parasites quickly travel to the liver, invading the liver cells called hepatocytes. Here they undergo rapid asexual reproduction in a process known as exoerythrocytic schizogony. Within about a week, thousands of new parasites called merozoites are released into the bloodstream where liver cells rupture. For some of these species, such as Plasmodium vivax, these can form a dormant stage called hypnozoites, which is important to note because they can reactivate months or even years later, explaining why malaria can relapse long after initial infection. So the next phase occurs in the red blood cells. Merozoites invade red blood cells and dramatically remodel them to support parasite growth. Inside these cells, the parasite progresses through recognizable stages: ring-shaped trophozoites, developing schizonts, and then finally they'll release more merozoites. This repeating cycle destroys red blood cells and produces the classic symptoms of malaria, including fever, chills, fatigue, anemia, and in severe cases, organ failure or death. Some parasites instead develop back into gametocytes, allowing transmission to continue when another mosquito feeds, completing the life cycle loop illustrated in this piece. Malaria is really difficult to control because Plasmodium adapts metabolically depending on its host. In mosquitoes, it relies on mitochondrial energy pathways, while in human blood, it depends heavily on glucose metabolism and digestion of hemoglobin. This flexibility, combined with drug resistance and mosquito insecticide resistance, makes prevention and treatment an ongoing challenge. So my artwork depicts that this is a continuous biological cycle connecting environment, insect, and human host. Global organizations such as the World Health Organization aim to reduce malaria deaths by 90% by 2030. Continued research, prevention efforts, and public awareness remain critical if malaria is to ever be eliminated. Thank you.
Eukaryotes dendropatitis. However, it has pretty quickly become one of my least favorite microorganisms. While it's very interesting, I do not like the effects that it's had on the world, unfortunately. I'll be telling you guys about it through a silly little story about some frogs. Let's do spooky campfire stories. Yeah! No! I have one, but I'll have to warn you. This is more than just a story. It's real. I'm talking about Batrachochytrium dendropatitis. What? We'll call it BD for short. This strange organism is a fungus. It's part of the Chytridiomycota division, also known as chytrids. These are all unicellular, but they are one of only 2 chytrids that are parasitic to vertebrates. In fact, they're so unique that they're the only members of their genus. They're part of the order Rhizophidiales, family Incertaceidis. Yawn. This isn't even scary. Get to the good part. Calm down, guys. Did you miss the part where it's parasitic? To us? These little microorganisms start as little zoospores that can move around. They then turn into sporangium that can't move anymore. Then they get bigger and bigger, and then they pop, releasing even more zoospores to go around and infect even more frogs. In frogs, it can live in 2 ways. Zoospores can burrow deep into the skin and then grow into sporangium in the endobiotic cycle. These then pop and spread the zoospores even further. Or it can follow the epibiotic cycle, where they stay on the skin surface. However, this is less common. If you get infected, lots can happen. You'll stop eating, you'll be tired all the time, your skin will turn gray or white or tan or red, and you won't be able to turn yourself right side up. You'll get chytridiomycosis, which messes with the osmoregulatory of your skin, and eventually, you'll die. It lives by eating your keratinized tissues in your skin. They can spread through infected soil or water, or by frogs getting too close to each other, and then it can get even further as humans trade us around the world. They're still figuring out all the specifics, but they think other species like crawfish can carry it around and spread it. Some frogs can get it with no issue, but then it can get spread to a frog that's never seen it before and make them super sick because they don't have any immunity or resources against it. It was first seen in Australia in the near tropics, but people think it actually comes from Korea originally. They don't know the exact start. This is a heat map of all the places they found it so far from all the frogs that they have tested in the world. And now it's spread all over. It's been found in 86 countries and counting. These are just the ones that they've tested frogs at. It infects all different kinds of frogs. Did you know that we're in an extinction crisis right now? Up to 50% of the frog species are at risk. Uh-oh. BD can infect all amphibians, from frogs to toads to newts, salamanders, and caecilians. But frogs are the ones who are most impacted. What was that? Your story sucks. I'm scared. Don't worry, guys. Domestic frogs can just get antifungal treatment. Yeah, but we're not domestic. I'm sure we'll be fine. We're hiding all the way out here. Dun, dun, dun. Thanks for listening, guys. That's all I have.